In the nutrient-poor bogs of the Carolina wetlands, evolution took a dark turn. Here, the soil is acidic, waterlogged, and almost completely devoid of nitrogen. For most plants, this is a death sentence, but for one species, it was an opportunity to rewrite the rules of biology. This is Dionea muscipula, the Venus flytrap. Charles Darwin famously called it one of the most wonderful plants in the world, but even he struggled to understand how a plant, an organism with no brain, no nerves, and no muscles, could hunt animals with such terrifying speed. The existence of this plant challenges our definition of intelligence. It does not just react, it calculates. It does not just move, it hunts. But how did it evolve? Genetic analysis reveals a shocking truth. The flytrap was once a passive sticky plant, much like a sundew. Over millions of years, it repurposed the genes used for root nutrient absorption and turned them into digestive enzymes. It took the genes used for stress defense and turned them into attack sensors. It evolved from a victim into a predator. And the mechanism it uses to catch prey is so advanced, so mathematically precise, that it remained a mystery to science for centuries. Welcome to the mind of a killer without a brain.
To the naked eye, it is just nature. But look closer. It is engineering. It is economics. It is survival. Welcome to the hidden side of biology. This is modern botany. Let's dissect the trap. Inside the red, nectar-scented lobes, you will see tiny, sensitive spikes. These are the trigger hairs. They are biological switches, but they are incredibly sensitive. A displacement of just 3 degrees with the force of a single mosquito, 30 micronewtons, is enough to arm the weapon. But they are part of a sophisticated short-term memory system. If a fly lands and touches a single hair, nothing happens. The plant remains frozen. Why? Because the plant understands the concept of false alarms. A raindrop or a falling leaf could trigger a single hair. Closing the trap requires immense energy, and wasting it on a non-living object could lead to starvation. So, the plant waits. The first touch generates a small electrical charge, an action potential that travels across the surface of the leaf. But this charge is sub-threshold. It starts a biochemical timer. The plant is now primed. It holds this electrical memory for exactly 20 seconds. If the insect touches a second hair within this 20-second window, the electrical charge doubles, crossing the threshold. Instantly, the cells on the outer surface of the trap expand, while the inner cells remain static, causing the leaf to snap convex. This is called thigmonasty. It happens in less than 1/10 of a second, faster than the blink of a human eye. The plant has counted to 2, calculated the probability of a live meal, and executed the strike. But the counting does not stop at 2. The capture is just the beginning. Once the trap snaps shut, it is not fully sealed. The cilia, the teeth on the edge, interlock like a cage, but there are gaps. Small insects can escape. This is intentional. The plant wants a substantial meal. It doesn't want to waste energy digesting a gnat. But if the prey is large enough, it panics, it struggles, and in doing so, it touches the trigger hairs again. 3 touches, 4 touches, 5 touches. At 5 touches, the plant releases a hormone called jasmonate. The trap begins to seal hermetically, pressing the edges together until it transforms from a leaf into a green stomach. Then, the chemical warfare begins. The plant releases an enzymatic cocktail containing acidic proteases. The trap becomes an acid bath with a pH of 2.0. It dissolves the insect's soft tissues, breaking down proteins into nitrogen and phosphorus. The more the prey fights, the more it stimulates the hairs, and the more acid the plant releases. The struggle for life only accelerates death. The biological complexity of Dionea makes it highly desirable, but unfortunately, this demand has led to poaching. The Venus flytrap is now endangered in the wild,
but you can save it by cloning it. Propagation of this plant is a lesson in totipotency, the ability of a plant cell to differentiate into any other type of cell. The secret lies in the rhizome, the white bulb-like stem hidden underground. There are two main surgical methods. First, leaf pulling. You must carefully peel a leaf downward, ensuring you detach a piece of the white rhizome tissue. This white zone is rich in meristematic cells. If you plant the green part alone, it will die. You need the white heart. Second, flower stalk propagation. Flowering is exhausting for the plant. Many growers cut the stalk to preserve energy, but this stalk is packed with growth hormones. By cutting it into sections and planting them vertically in sphagnum moss, you can trigger vegetative apomixis. Within weeks, the undifferentiated cells will reorganize, forming tiny, perfect clones of the parent. You are not just growing a plant, you are replicating a genetic masterpiece.
A memory without a brain, movement without muscles, digestion without a stomach. The Venus flytrap is a reminder that intelligence is not the exclusive property of the animal kingdom. It is a sentinel of the swamps, counting the seconds, calculating the odds, and teaching us that in the game of survival, patience is the deadliest weapon of all. This is not just a plant, it is an alien intelligence that grows from the mud.